Welcome. Welcome to Pharmaceutical Analysis session. I'm Dr. Ahmed Nawaz Khan, and today we are going to talk about the definition and scope of this pharmaceutical analysis. Imagine yourself that you are in a grocery store and you just select your favorite food lunch. For example, you pick up a jar of peanut butter, look at the label, read that label, and you find that there is 190 milligram of sodium is there in one serving. What do you think? You think that how the product knows that it contains 190 milligram of sodium. Just after that, you just again pick another item, and this time you pick a toothpaste, and then you find that the label contain that fluoride content is 0.15% weight per volume. Then again you were surprised that how they know that there is a fluoride content in the toothpaste is 0.15. Now again you go to move to the other section, and you find a vitamin bottle, which is the multivitamin bottle, and you find that there is mentioned that 1.7 milligram of riboflavin in every tablet. So what do you think that how the manufacturer they come to know that how much content is there inside the product? The same way, there are a number of tablets, there are a number of dosage forms which have the labels, and on labels the drug name or the excipient name, they are written with their content. So this is the science which tells you about that what is the content inside the product and how much the quantity is. The science which tells you that how much the content, what type of elements are there, what types of element are there, what types of chemical compounds are there inside the products, and what is the quantity of that products or the chemical compound in the product. It will tell, the analytical science tells you this. So now let's move to the definitions. What are the definitions of some very important definition which are related to this pharmaceutical analysis? So the first is analytical science. What is analytical science? Analytical science is a science that deals with the identification, quantification of the components of the material system. And this science is known as the analytical science. It involves the identification as well as quantification of the compound or the components. This is called because the process of determining the level or any compound or all the compound in the material system is known as the analysis. So the science which deals with the identification and quantification of the compound, this is known as the analytical science, and the process of determining the level of one or more than one or all the compounds in a material system is called the analysis. This analysis, it involves both physical as well as chemical process, because analysis is a process. So it involves both the physical and the chemical process. Now, if the chemical is involved, it is known as the chemical analysis. Or we can broadly say that this chemical analysis is known as the analytical chemistry. Now, whether you are looking for the sodium in the peanut butter, the nitrate in the water, or ozone in the air, the scenario is that you want to find out that which compound, which component is there, and at what is the level of that component or compound is there in the material system. And the process of knowing, that means identification and quantification of the sodium, ozone, right, is known as the analysis. Now there is a word which is known as the analyte. The substance which is used to be identified or which is used to be quantified is known as the analyte. And the analyte is present in a matrix system. Matrix is a bulk form which contain the analyte, that means component of the compound, plus some extra material which give them it a bulk form, just like in the case of the pharmaceutical products. In the analysis, you will hear this name so many times. You will see that the terminology is standardization. What is the standardization? Basically, it is used while you are doing the titrations. We'll discuss it in the coming classes, in the coming session. What is the titration? Now, what is the standardization? When we prepare a solution, when we prepare a sample, right? We'll discuss that what is a sample. Then we want to confirm the of that sample, and that is known as the standardization. This standardization has different meanings, but they are all a similar kind of meaning. The next is the assay. What is an assay? That when we used a compound and we make a product, that compound has some purity level. That how much compound is pure, right? So this is to determine the level of the purity, and that purity is known as the assay value. Now the next is the characterization of the material. So why we are using this analytical science or these methods which are there to analyze to do the assay or the standardization? Because of the characterization of a material or a product. So a combination of qualitative plus the quantitative. As the name suggests, the qualitative, that means it is a quality parameter, and while the quantitative is a quantitative parameter. So the combination of the qualitative as well as the quantitative analysis of a material or a matrix is known as the characterization of the material. And the characterization of material overall tells you about the quality of the product. Now, for the number of techniques, number of methods, equipments, process, all these encompasses the analytical sciences. Now let's discuss about the classification of analysis. How the analysis is classified. Now see the procedures which are used in the analysis, they are classified broadly into two ways. The based on the goal of the analysis, and the second the so when we talk goal of the analysis, the classification can be on whether the analysis is qualitative or quantitative. When we are talking about goal of analysis, so the analysis is qualitative or the quantitative. What is this qualitative analysis? Basically, it is the identification. If we say in other words, it is the analysis which is carried out to determine only the analyte, the identity of an analyte in matrix, or the identity of several or multiple compounds of a mixture. Now, the another is analysis to determine what a material is or what the component of a mixture are. So such an analysis does not require or does not need the amount of the substance. Chemical analysis carried out and it is reported that, for example, there is mercury present in water in a lake and the quantity of mercury is not reported, then the analysis was a qualitative analysis. What it means that when the values are not given, when the exact values are not given, whether the identification, so this is known as the qualitative analysis. Now, what is quantitative analysis? So, quantitative, on the other hand, is the analysis of material, how much of one or more component is present. And such type of analysis, they are undertaken when the identity of the compound is already known. And when it is important also to know the quantity of these components. So it is the determinant of the quantities of one or more than one component present in the sample or in the matrix. Now the analysis process, they are also additionally classified into procedures that involve the physical properties, wet chemical analysis procedures, instrumental chemical analysis procedures. So broadly they are qualitative, and additionally they are classified into procedures involved the physical properties, wet chemical and the instrumental analysis procedures. So just like analysis using the physical properties involves no chemical reaction, and at times it is relatively simple devices, although there are some computerized, right, to just create the measurement. Physical properties are especially useful for application, but may also be used for quantitative analysis, right? Just like in the case of specificity or refractive index, etc. So, and they vary with the quantity of the analyte in the mixture. What is wet chemical analysis? It is usually involves the chemical reaction. The classical reaction is stoichiometric, but there is no electronic instrumentation is used beyond a weight device, except the weight devices. So wet chemical analysis take very classical technique, meaning that they have in the analytical laboratory years before electrons came on the scene or used. If executed properly, they have a very high degree of accuracy and precision. Will come in the coming session what is an accuracy and what is precision, but they take more time to be executed. Now, what is the instrumental analysis? This instrumental analysis can also involve the chemical reaction, but it always involves modern sophisticated electronic instrumentations. Instrumentation analysis technique, they are highly high-tech techniques which are the ultimate in a very complex hardware and softwares. Sometimes not as precise as a carefully wet chemical method. Instrumental analysis method, they are very fast and they can offer a much greater scope in the practical of the analysis. So this in addition instrument methods are generally used to determine the constituents or the constituents which are present in a very low level rather than some major constituents of a sample. So this is about the analytical techniques, but the more appropriate is classification of analysis, because techniques, there are a number of techniques which we will learn in sessions. So this is basically the classification, or you can say broad classification of the analysis. So now this sample, this is very important because whatever we are going to analyze, we need a sample. We need a sample to be analyzed. This is the term for the material which is under the investigation, and this material can be single material or this material can be in the bulk system. That means it contains some of the other components, and that's why this combination of the components, so the bulk system or the matrix is the case of analyzing, just like the toothpaste for fluoride in the toothpaste. So there are enough components there, like the tablets. There are a number of pharmaceutical active ingredients are there, number of excipients are there. So they form a bulk system, right? So when we are going to analyze, we need a sample. The single sample or pure sample you are going to analyze. You want to know the standardization of that sample, or there is a samples which is a matrix. So now what is the native samples? Actually, this is the, because we are studying the analysis. So a representative sample, that means the sample which represent the whole batch. For example, you know these agriculture, the scientists or the researcher, they what they do, they collect the sand or the soil or from the deep for analyzing the sample of or the soil, right? They do not, they choose where there are more chances to be of component which they want. So they choose a very specific location, but they never get biased for that location, but they choose a very important location, and that portion of the matrix or that portion is known as the representative portion or the sample. This sample represent the whole land, because it's quite tough to get, tough, impossible that you take the whole land, take it to the laboratory, right, and then analyze the land. No, you have to take portion of the land, that is known as the representative. In the same way, if there are number of for example, a batch of 5 millions of tablets are there, tablets you need, for example, you need at least 20 tablets, and these 20 for the analysis is known as the representative portion of that tablet. And this is overall, they are known as the samples. Now next is the analytical strategy. While you are working on a sample, you are working in a laboratory, you must have an analytical strategy. That means the strategy through which you start your work and you will end your work successfully. So the process by which the analyst analyze the, or means identify or find out the concentration level in a sample, involve many individual steps. So for steps, they are, the first step in this is obtain the sample. When you are obtaining a sample, you must make sure yourself that obtained by you is appropriate. That sample or the claim sample, this only. The obtained sample must be stored at a proper place, and when you are using it make sure that it's expired. The product must be in very the life. Now when you preparing the sample, there are a number of techniques which are used. So when you are getting the one very important which I forgot about, the source is very important. Write down source of the sample you are getting, the place, manufacturer, from where any source from where you are getting the sample. The second is preparation of the sample. That means when you start preparation of the sample, how much volume is required, what solvent you are requiring, right? What is the which chemical you are requiring to make the sample, right? And which glassware you, right? It must be calibrated glassware. So the preparation of the sample. The third one is the analysis method. That which analysis method is appropriate for that sample. This is the basic main strategy while you are working with the analysis. That which method to analyze the sample, right? Just like, I'm not sophisticated kind of analytical methods, just I'm talking about just very simple methods, just like the titration method. If a sample is not if it is an acid or base and it is not dissolvable in water, then we use the non-aqueous titrations, right? So the method of analysis is very important, choosing or carry out the analysis method. Now thereafter, when you get up with you have done with the analysis method, you start to collect the data which is coming which you will get from the your samples, right? After getting the data, just analyze the data, sit with the data and think that is it the right which you want, or it is the right data which you get, right? Then start calculating and then finally report. Try to manipulate the data, because it's a for a pharmaceutical analyst, that the manipulation, they many times manipulation is a very unethical. So never do the manipulation of the data, whatever the data, whether it is positive or negative. So you must write the same data and you will calculate with that data only, right? So this is how the analytical must use, or this is the approach. You first find obtain the sample, the source of the sample, right? It is must be the right sample. Then we start the sample in the preparation instrument, what type of glassware you are using, what type of it must be calibrated, and what chemicals you are using. Then carry out the analysis, which analytical method is best for the analysis of the sample, given sample, what is the nature of the sample. Basically it is based on the nature of the sample, and then work up with the data. When you get the values of the data, then you just think calmly and then think about the data, and then start calculate formula and softwares and etc. And then thereafter report, and the report as such, or the values you are getting from the your results, right? So do not report, do not conclude anything or manipulate your data. This is very important while you following the good analytical strategy. Now techniques and skills, right? So after approaches, who adopt the good approaches or good strategy for the analysis? The one who knows the technique, the one who knows the skill, the one who have the skills. Now, so we must, a student or any analyst must have good analytical technique, follow the good analytical techniques, and must have the good analytical skills. Now, for example, every person has a special mindset, special skills. So before working on the sample, he or she must be a thinking person, a person who pays close attention to the details and never get it off, never get it off, just to for getting the high quality of the data and the result while you are working in the laboratory. So these kind of skills and the skills which you will learn in the laboratory by the the weighing, working with the washing of the glassware, rinsing of the glass, etc. So these are the good analytical skills, right? And the skills, when you will start in the advanced techniques, so the skills are different, right? So you must take great care of your lab, your sample, associated material. The sample should be contaminated. This is the best practice which you do while you are working in the lab, working with the sample. The contamination, never try to save it, at a proper place. Always use some the standard conditions to store the sample, and so that when you are using the sample or the chemicals, your skills will definitely avoid some accidental issues, right, which then happens right in the pharmaceutical lab, because you all guys, we are chemicals, and but the chemical, the nature of those chemicals, but the chemical does not know, right? So it is better that you all your care with the all your knowledge, that what is the chemical, and you start your working in the analysis of the sample. So the last I want to say that when you are start working, first stop and think before you proceed with a new step in the procedure. What might happen in this step that would cause contamination or loss of the sample? These are the points which you have to think, right? If I keep this sample in here, what will be the contamination? What will happen? If I do this, what will happen? Don't follow that thing, but only just think about it. What will happen if I do this? What will happen, right? So this is all about the analytical techniques in skill. Now, after skill you will learn in the practical classes, and the techniques definitely you will also learn throughout this semester. We'll do the titration techniques, different types of titration techniques, and we'll perform in the lab, right? So this is all about for today. So see you guys in the next session.